Hi there, this is Will from the Translational Neuroscience YouTube channel, and today I'm going to be covering a very fundamental topic in neuroscience, and that is the concept of the resting membrane potential. And today we are going to be going over the basic concept. In subsequent videos, we're going to go a little bit more into some of the details, some of the finer points, some of the mathematics that govern how this resting potential works. But for today, we're just going to go over the really basic concept. So the neuron, if you'll recall, has three parts. We have the cell body, which has the nucleus. It's kind of the command center of the cell. We have the dendrites that receive information, and then we have the axon, which generates action potentials or electrical impulses that propagate down the axon, and those in turn will allow the cell to communicate with downstream cells. And in order to understand how this impulse works, we have to understand how the membrane that propagates it works. And so if we were to zoom in on a little portion of that membrane, what we would find is we would find a double layer. And this is going to be your a membrane, much like the membrane of many other cells, where you have it's comprised of phospholipids with a hydrophilic head and a hydrophobic tail. And on either side, we're going to have cerebrospinal fluid, which is basically water with some ions in it. And there are two types of ions, right? We have positively charged and negatively charged ions. Positively charged ions are referred to as cations, and some of the ions that we're going to encounter here are the sodium ion, the potassium ion, and the calcium ion. And some of the negative ions that we'll encounter, or anions. An example of an anion that we'll encounter is the chloride anion. But we're not going to talk too much about these specific anions and cations today. Today we're just going to talk about the very basic concept. So the basic concept here is the concept of electrostatics. So positive ions and negative ions attract each other, and like ions, so a bunch of positive ions together are going to repel each other. Same goes for negative ions. So on one side of the membrane, so this is going to be outside the cell, and this is going to be inside the cell. Outside of the cell, we have a bunch of positive and negative ions. Inside the cell, we also have a bunch of positive and negative ions. We'll go into in later videos what those ions are. But suffice it to say that on the inside of the cell, there's an excess of negative charge. So we have more negatively charged ions on the inside than on the outside. And what that is going to cause is that's going to cause positive ions on the outside of the cell to become attracted to this membrane. But remember, these two types of ions can't get through the membrane because this membrane is impermeable to ions except under certain special conditions that we'll go over. And so what this creates is a situation where you have potential energy. So if we were to get rid of this membrane, there would be a flow of current, a flow of ions. And since this situation is occurring at the membrane, we call this the membrane potential. And at rest, since we have more negative ions on the inside than on the outside, and we always define the membrane potential by convention to be inside with respect to outside. That's the convention that we use. So at rest, the membrane potential is negative on the inside with respect to the outside, and a value that is useful to know is it's usually around negative 70 millivolts is that value. And so this is a situation where the membrane is impermeable to ions. Ions can't move through the membrane, but there are going to be certain situations in which ions actually can move from the inside to the outside or vice versa of the membrane. And we'll go into what the physical basis of those ionic movements are later. But today, let's just consider a situation, hypothetical situation, where our resting membrane potential, Vm, is sitting around its regular value, which is about -70. And then at a certain time, Moses, so we'll call this T Moses, is going to come along and part the membrane. And he is going to say, I'm going to create a channel, and this magical channel shall allow only negative ions from the inside of the cell to emerge, to go through the membrane to the outside of the cell. And you say, okay, well, that seems pretty reasonable, right? Because we have more negative ions on the inside and more positive on the outside, then the negative ions are going to be attracted to the positive. They're going to want to get out of the cell. So if Moses creates this channel, that's a pretty natural thing to happen. And so we've got our time elapsing here. This is time on this axis. Time is elapsing, and then Moses comes in, and since negative ions are moving out of the cell, the difference between the potential on the inside and the outside is going to be less. So this membrane potential is going to become less negative, right? It's going to move in this direction. And then at some point, Moses is going to close the channel, and the membrane potential will be less negative at that point. And that process of the membrane potential becoming less negative is something that we call a depolarization. And the opposite could happen too here, right? So we could have a channel that allowed positive ions to come into the cell, and that would have the same effect. But say Moses opens this other channel, and he says, actually, that channel didn't happen. We're going to open a different channel, and this channel is going to allow positive ions to move out of the cell. And you say, well, wait a minute. That doesn't make a lot of sense, right? Because we have more positive ions on the outside than on the inside. Why would positive ions want to go to an area where there's more positive ions? Doesn't make sense, violates the laws of electrostatics. But what we're going to see is that there are actually two driving forces at play here. Number one is the driving force that we just talked about, which is the electrical potential. Electrical potential. But then number two is actually the chemical potential. And we will go into this in a subsequent video, but suffice it to say that ions actually can move in a direction of, so a positive ion could go to an area where there are more positive ions if that movement was favorable for its chemical potential. And likewise, negative ions can do the same thing. And I know this is a little bit confusing now, but we're going to go over it later. And so that movement, so what would that do? Well, if we have positive ions moving out of the cell, that is going to mean that there's going to be more positive charge here, which means that this is going to become more positive relative to this, or this is going to become more negative relative to this. So what we have is the opposite effect, where the membrane potential becomes more negative. And this is called a hyperpolarization. And under most situations, these little changes in membrane potential that are happening all the time are not going to result in any big change. So as long as there's a depolarization that happens that's going to be less than a certain threshold, then nothing really happens. There's no action potential, nothing dramatic happens, and we call those electrotonic potentials. But if that depolarization were to go past this threshold, then we would see this massive fluctuation in the in the membrane potential, and those are those action potentials that we talk about. And we are going to go into how those work in a subsequent video. So that is the very basic nuts and bolts of the resting membrane potential. And in subsequent videos, we are going to go into more detail about how this is working and how the chemical and electrical potential are interacting to create this, this membrane potential. So thanks.